Welcome to the NeuroExpress tutorial video. NeuroExpress is a Windows program designed for the analysis of electrophysiological data acquired in patch clamp experiments. These data include voltage responses of neurons receiving current step stimulation. Also, the program can analyze spontaneous or miniature postsynaptic excitatory or inhibitory currents, typically recorded in gap-free mode of acquisition. And the third type of task the program can perform is the analysis of spike trains or voltage responses of neurons firing spontaneously under network inputs or chemical drive or electrical stimulation. Today I will mostly talk about the analysis of current step responses. So when neurons receive current step stimulation under whole cell patch clamp conditions. You can download the trial version of the NeuroExpress program from my ResearchGate page. You can locate this page by googling NeuroExpress mini-analysis. And the second link here will take you to this download page. Here you can download the program as a zip file, so a compressed archive that contains the executable, a brief documentation, and also some sample files that you can work with or you can test out. When you download the program, it will run as a trial version, so a fully functional program for 50 days, and then it will turn into demo mode. But I encourage potential users to visit this ResearchGate page on a regular basis because I upload newer versions of the program as I develop it. So the 50-day trial version will restart when you install the latest version. So how to do that? It's very simple. You can create a folder in your computer like C/NeuroExpress, and then you can copy the executable, which is called neuroexpress.exe. There is also an image or animation creating utility nextgif.exe, and the sample files can be also put here or maybe into a separate subfolder. So this is very simple. You do not need to download any runtime libraries or Python environment for running the program, and it can be started just by clicking on here. And this is what you will see, and then you will be ready to use it. To analyze the voltage responses of neurons under current step stimulation, you perform a whole cell experiment in episodic stimulation mode. Here we record the injected current that was used to stimulate the neuron, and of course the membrane potential response of the neuron. So these are two-channel recordings, and the acquisition mode is called episodic stimulation. Typically, what we want to achieve is that we start at the most negative current level. In this case, it was minus 150.7 picoam, and then we incremented this injected level in small steps in order to gain a number of voltage responses from the neuron. So some of these responses are hyperpolarizing, and then when the neuron receives positive currents, then it will be depolarized and start emitting action potentials. So this is a very useful protocol to investigate a number of physiological parameters and intrinsic biophysical parameters of the cells of interest. We can calculate the membrane resistance, membrane time constant, voltage, sag, reobase, and many more. What kind of files we can actually load? What format of files we can load? So the most important is the Axon binary format. And this is the format that the P-clamp electrophysiological or patch clamp suite software environment produces. So ABF files are compatible. Also the Axon text file, and there are a few other file types that I can describe later. To open a file, it's very simple. You click on the open button here, and then you can open your target file that you want to analyze. So in this case, the name of the file already tells that there are some IV data in this file, and I open it, and you can see that the analysis was also performed very quickly and efficiently. So on the top panel, as I already showed, there are these aligned overlapping voltage traces from the neuron. The entire experimental recording will be shown in the middle panel as full trace, so the membrane potential is a function of the elapsed time. But the most important plots are below, and here you can actually select what you want to see. For example, in this case, we would like to display the voltage sag as a function of the injected current, and I click here. So you can access all these functionalities by right mouse button, clicking the right mouse button, or by clicking here and bringing up this little pop-up menu. You can also see that when I move the mouse cursor, then the voltage trace associated with the calculated parameter will be shown on the top graphical panel. When I move the mouse cursor outside, or just left to the opposite of the panel, then all the recorded traces will be shown. But also you can zoom into your data and just go through using the scroll function here. You can also change the Y scale here, which is useful to look into the very tiny details. So if I click auto rescale the Y-axis on the voltage trace panel, then these traces will be shown in the maximum magnification, like here. In this case, you can see that the cell receives some excited postsynaptic potentials. As a result, the trace is not very smooth, and there are these transients. Actually, this can change, or this can affect the accuracy of the analysis, and I will describe this very soon. So let's see how the program will work to extract physiological parameters from these IV data, from these voltage responses of the neuron. In this example, the neuron received a stimulation starting at minus 153 picoamp, and the total number of steps was 61. So there was a strong firing at the polarizing range. Membrane resistance is one of the most important parameters, and the program will use two different methods basically to calculate the membrane resistance. The conventional method is based on the linear fit of the voltage versus current relationship. So here the program will first look for the most negative and most hyperpolarized voltage level under the negative current step, but it will also look for the steady state voltage membrane potential level at the very end of the voltage step, or the current injection. So here we will have a baseline here, or we can also call it resting membrane potential, the most negative voltage deflection, and the steady state voltage deflection. And based on these different values, we can see the black and gray trace. So these are basically the membrane potential levels displayed or plotted against the current, and the slope of these relationships will be used to estimate the membrane resistance at I equals 0, that is when no current is injected. So the black trajectory will yield 272 mega ohm. That is what we calculate from this linear fit. And then the next method is slightly different, and this is actually what I prefer to calculate the resistance in a more accurate manner. Here, the program will calculate a delta V over delta I value, resistance value for each voltage trace, each membrane potential response. As you can see, these delta V over delta I values are not just a single constant, but they range from 250 to 300 or even 350 megaohm. So they are dependent on the current level, the injected current that was used to evoke the voltage response. We can use a linear fit to extrapolate these values to I equals 0, so to obtain the membrane resistance at 0 current level, that is the resting membrane potential of the neuron. In this case, the estimated membrane resistance would be 317 megohm instead of 272. Also, the resistance will be different for the green points which are extracted from the steady state voltage deflection. So delta V is smaller for these green steady state values than for the maximum values. So this is kind of expected when we have such a strong voltage sag. Voltage sag is basically a depolarizing change in the membrane potential during negative current injections, and this is very common in a wide variety of mammalian neurons. This behavior is caused by the action of the hyperpolarization activated non-specific cation current, or IH. So whenever IH is strong in a cell, we can expect a behavior like this. And the voltage sag can be characterized by voltage sag and rebound versus current relationship, as shown here. So basically, we are just plotting the voltage sag as a function of the injected current. So these maroon colored symbols will show how much the membrane potential will depolarize after the maximum. So how much is the difference between the voltage at the green and the red locations. And this will also depend, of course, on the current, on the injected current level. So the relationship between the injected current and the voltage sag is usually linear, but sometimes it flattens a little bit. But we can actually fit a very nice and clean line here, as you can see the gray line, and that will give you a slope parameter for the voltage sag versus current relationship. There is the second relationship indicated with these olive colored symbols. That would be the rebound potential or after depolarization. So how we interpret that? When the current step ends and the current goes back to 0, then the membrane potential can overshoot. Basically, it can reach a local maximum that is more depolarized than the baseline, the resting membrane potential. So the difference between the overshoot and the resting membrane potential will be used to calculate the after depolarization ADP. It can be also called rebound potential. In this case, the rebound potential is slightly lower than the voltage sag, but quite often it is overlapping with the voltage sag trajectory. We can also calculate the relative voltage sag. This is a percentage of the sag, this sag potential relative to the maximum voltage deflection. So basically we are dividing voltage sag here with this delta V, and that ratio will be expressed in percents. So in this particular example, the relative voltage sag or sag index is extremely high, more than 40%. The after depolarization can be also expressed as percentage. So basically we are dividing the ADP with the maximum voltage deflection. And that's what we display on this plot. So these are all very useful parameters, and measures of the physiological behavior of the cell, and we can use the program to calculate these very quickly and efficiently. In this example, I will show you how the program calculates membrane time constant. Well, basically, when the membrane potential start getting more hyperpolarized during the negative current injection, there is a mono-exponential onset of these voltage traces. And the program will fit a mono-exponential function automatically based on the onset time, or I mean the start time of the current step, and also the location of the maximum voltage deflection. So the program will look for the most exponentially looking part of the voltage traces and perform a nonlinear fit. Like here you can see these magenta traces nicely overlapping with the actual biological response. So the time constant, or the tau, calculated from these traces will be shown as these magenta dots. So in this case, you can see that the onset gets a little bit slower and slower. The time constant will rise as we are getting closer to I equals 0. So these magenta points that will depict the estimated time constant for the particular voltage trace, they will also depend on the injected current, and it can be a huge difference between negative or most hyperpolarized responses versus the response very close to I equals 0. In this example, the time constant ranges from 10 to 45 or even 50 milliseconds. So again, it's probably a good idea to use linear regression to estimate the behavior of this relationship. And then you can see this little rectangle, which is the extrapolated time constant at I equals 0. And the program will report 49.7 milliseconds for the time constant at 0. The program will also attempt to fit the decay phase of the membrane potential and it will depict these traces in bluish color, and these blue dots will show the time constant values extracted from these ones. Also, when we have time constant and resistance calculated for the different voltage traces, then we can use the estimation of the membrane capacitance from these responses, and we can bring up this this parameter by clicking here, estimated capacitance. So as you can see, the capacitance, unfortunately, it is also a little bit dependent on the membrane current. Unfortunately, I say, because membrane capacitance should not be a function of the current, but we have to admit that there are multiple or a multitude of ionic currents playing here. For example, in this case, the neuron exhibits a very strong inward activation, and there are inward actifying potassium channels driving the response of the neuron. Indeed, the membrane resistance seems to be only 200 picoamp at the most negative current step, but close to 0 it rises to 500 megaohm. So there is a very high degree of inward rectification, and this rectification will affect the membrane time constant and the estimated membrane capacitance. So we can definitely try to extrapolate these points and to get a reasonably good estimation of this capacitance at I equals 0, so at resting membrane potential. What else can we show here? Of course, the program will also analyze the input-output curve of the cell. In this case, the neuron started firing around 30 picoamp. Yes, 30.7 picoamp with a single action potential, and then the number of action potentials raised. You can see the program is doing a fairly good job finding all the spikes and determining the threshold of the voltage threshold of the firing. The green points here will indicate the most negative membrane potential level reached between spikes. So this value can be used to calculate the after hyperpolarization, spike after hyperpolarization. All the spike parameters can be accessed here, for example, spike amplitude. Here, the amplitude values are shown for the first, second, third, etc. Number of spikes, and the y-axis will show the amplitude in millivolts. So let me show here. So these are the big spikes. The first spike is always bigger than the second and the third because there is a little bit of spike amplitude adaptation. So here the amplitude will go down as we reach stronger and stronger current levels. We can also display spike threshold. These values usually go higher and higher, so the membrane potential or spike threshold gets a little bit more and more depolarized, and you can see these gray points, they are also walking up. So that means that the threshold membrane potential gets higher and higher along the current step. This is also normal, and most neurons exhibit this behavior. We can calculate spike half width and fall time, and then several other parameters. As I mentioned, there is also after hyperpolarization, which is a positive number for spikes when the threshold is higher than after hyperpolarization. So these are after hyperpolarization values calculated for the first, second, and other spikes. In this next example, I would like to show you how to set different parameters to optimize our analysis of the IV traces, IV data. So these numerical parameters can be accessed by clicking and editing these numbers here. And you can also use the mouse wheel to increment or decrement these parameters. So this is a function that can speed up your work tremendously because you don't need to use the keyboard. You can use the mouse just to increment or decrement these numbers. Anyway, let's start with the current reject range. Basically, this is a parameter that will be used to optimize the calculation of the membrane resistance, but it will also affect that membrane time constant. So when the program calculates the membrane resistance, it will divide the delta V over delta I. So the delta V is basically the voltage deflection under the different current steps. But when we are getting very close to the zero current step, basically the voltage deflection becomes very small. Yes, like here it's very small. And the current will also gets very small. So we are dividing basically a small number with another small number. Unfortunately, the membrane is never really smooth, and the membrane potential is never really perfect. There are sometimes EPSPs riding on top of the trace, or maybe some intrinsic membrane fluctuations or intrinsic noise that contaminates this. As a result, the noise can change the observed maximum voltage deflection by a few millivolts, or maybe just less than 1 millivolt, but basically the contribution of the noise to the calculated membrane resistance becomes stronger and stronger. That is very much true close to I equals 0. So basically, if we want to remove these points from the calculation, then we can use this current reject range parameter. Basically, from minus 10 to plus 10 picoamp of current stimuli, we are not calculating membrane resistance at all. But if I want to increase this range, then I can go to 20 or even higher. So you can see that points will disappear from the calculation and also from the membrane time constant estimation. So this can remove those points that will contaminate the membrane resistance too much, and maybe it would degrade the accuracy of the estimation for membrane resistance. The other next point is resistance midpoint. This is a different thing. Basically, when we use linear regression to estimate the membrane resistance, we can tell the program what would be the most negative current level from which we draw the line up to 0. So I can decrease this number and I can go to very low negative currents, or I can go to a little bit more depolarizing currents like here, and then the program will recalculate these linear regressions. I can turn off or on the double linear fit feature for this, so we can have a second line for the resistance curve. Also, this is true for the voltage sag, and we can change these parameters with the mouse wheel. So if I decrease this, then I can make a very long linear regression. Okay, so these are basically midpoints of the linear fits, and these are very useful to get very clean linear fits where it is appropriate. When we have the next parameter, input-output spike number fit limit, let me show you how that works. So basically the program will try to fit a line to the points depicting the spike number as a function of the current. And this can be a fairly linear relationship for some neurons, but not always. And then we can determine up to which spike count we want to perform the linear fit. In this case it's 6, but I can decrease it to 5, 4, even 3 or 2, and then depending on this choice the fitting will get you different levels or different slopes. So the last parameter here is spike counting limit, and this is basically a parameter that will describe what is the maximum current up to which level you would like to accumulate or sum up all the action potentials. In this case it's 150, so basically the program will find the spikes, will calculate how many spikes were emitted for the different current stimuli, and it will accumulate or sum up all the spike counts up to 150 picoamps. So this is a metric we are using for estimating the intrinsic excitability of the neuron or characterizing it. And this is a parameter estimating the cumulative spike count for different neurons. And of course this can be also different, but of course you would like to set this parameter to a level where you actually reach. So in this case the current went up to plus 200 picoamps, so I can set this parameter to 200 picoamps, but it would make no sense to make it 300 because I have no data at more positive currents than plus 200 picoamp. In the next example, I'd like to show how to perform manual correction of the data when it becomes necessary. In this example, the neuron was not synaptically isolated in an acute brain slice, and this neuron received EPSPs during the experiment. In this case, for example, there is a marked EPSP riding on the voltage signal, voltage trace, and the program finds the most depolarizing or most positive membrane potential level during the current step. The current was already positive here, 10.2 picoamp. So the program is looking for the most positive membrane potential. But in this case, the most positive membrane potential level is actually not the intrinsic behavior of the cell, but this voltage is caused by an EPSP. We can see that the membrane potential rises smoothly, and then it should probably just flatten or smooth out, but then there is this point here. So we can see that this is not the true maximum voltage deflection of the cell, but this is something that the EPSP caused. So how to help the program to deal with this little problem? There is a button here, automatic versus manual correction mode. When I click here, then you will see the correction core label in red appearing here. And now I can go to this point that I would like to correct and move to a place, a position where I think it should be. I click on it and I can drag it with the mouse, and you can see that actually the membrane potential rises here very nicely, and this is it flattens. So this is the location where the membrane potential should be instead of the top of the IPSP. Also, at the steady state level, steady state membrane potential level, this EPSP did not fully decay. And actually, I would assume or expect that the membrane potential will go to minus 68 millivolt or something like that. I can move this point down here. So now I corrected these 2 points. In addition to these points, the membrane potential was also a little bit shaky or a little strange here. And I am actually pretty sure that there was an additional EPSP that was just decaying before the current step started. So here we have an EPSP that did not fully decay before the step. You can also move this point up and down, and then of course the associated membrane resistance value will be recalculated. Let's see another example. This after depolarization value here seems to be way off. What is the reason? Well, it seems like when the membrane potential decays after the negative current step, we have a bunch of accumulating EPSPs. And the most positive membrane potential, so the overshoot, is indicated by this yellow symbol. I can grab that and move it somewhere here. I think it can be approximately here. So you can see that after depolarization is much smaller now for that particular point. Also here I can change that to or here, and we can perform these manipulations in order to smooth out the calculated voltage sag and resistance curves. Oh, here very close to I equals 0. Actually, this was almost 0. I can get down here, and the steady state is also somewhere here. So all these manual corrections can be performed. Of course, you want to be careful how you perform, and if you are not confident that you are doing the right thing, then you can definitely ignore this functionality. The best way to get beautiful, very clean estimation of parameters is that you get rid of the EPSPs and IPSPs in your recordings. So basically you are using synaptic isolation. So just to show an example, let's see some data here. Oh, maybe here. Yes, so this is a synaptically isolated cell, and there are no EPSPs and IPSPs here, and the membrane potential as a function of current is very smooth. The resistance and even the voltage sag is very nice and smooth. So we want to do these manual corrections carefully, but if you get familiar with this technique, that can improve your analysis tremendously. So after we set up all the parameters and optimized our analysis, we want to save the calculated physiological parameters into a result file or a worksheet. Results can be saved into an Excel worksheet if you have Excel installed in your computer, or into a comma-separated values file, which is compatible with Excel, or just a tab-separated text file. I usually save my data in Excel worksheet. To do that, you simply click on the Send button, and an Excel worksheet will show up, and it will show all these calculated parameters like here. So basically, the file name will be shown in the first column, and some metadata that you want to maybe fill out just to help your sorting of the data and to make the statistical comparison easier. And then you will see all these calculated parameters. For example, the resting current or holding current you applied during the experiment, the resting membrane potential of the cell, that is the average of the baseline points, and there are these calculated parameters for the membrane resistance. And some of these parameters or the headers are highlighted. These are the parameters that they are probably the most important and most widely used ones. Membrane resistance at zero current, so the input resistance of the cell is shown here. The steady state, the end point resistance is the second. Membrane time constant, membrane capacitance, the percentage or the voltage seg calculated from multiple voltage seg values, and the percentage of voltage seg relative to the maximal voltage deflection. So the voltage seg index or relative voltage seg is shown. The slope of the voltage seg response here, ADP, rho base, input-output gain, etc. So there are all these parameters, and the program will just send all these calculations into the Excel worksheet. So if you analyze several files, then you can do this by just sending, go to the next one, send again, go to the next one, send again. So basically the program will create this worksheet for you, and then you can compare these values for the different recordings, different cells that you recorded. And this is how it will work. Also, you can send these parameters to ASCII files. These text or CSV files will be created in the folder where the NeuroExpress program is located. If you made a mistake during the saving or the sending of the parameters, you can delete the last entry. So now the last line disappeared, and then you go to the next one and you send a different one, and then you can see that this will be filled nicely. So basically you can perform the analysis very quickly and efficiently. The program will not wait for the user input and instructions to calculate these parameters. Basically it will just do it automatically, but of course you might want to use the settings, change the settings and the slopes and these midpoints or the linear fits, spike counting limit, etc., in order to get the best estimation of the physiological parameters for the particular cell type or the cell that you recorded from. There are a few other things that you may be interested. You can export the traces. So if you have, let's say, an interesting behavior like the stuttering here, then you can export this, and basically you will get an ASCII file here. This is like the trace in readable format, that and this can be loaded into Excel or Origin or GraphPad or other program that you may use. Also, there is another functionality, creating animations, and I can show that. If you click here, then the program will basically run through the recording, and this little utility will show up. And this is the AnimGIF program or AnimGIF utility that will create animated GIF from this replaying of the traces. So in this case, the program will use 113 frames, 113 current steps, but it will go from the most negative to the most positive first, and then it will walk back. So this will take some time, because the total number of frames is 2 times 113. So let's wait until it's done, and I will show the result of that after it is finished. So the next GIF utility performed all the calculations and build this little GIF, animated GIF file. And I can show you here. There is this little image viewer program that will show what the animation looks like. But basically, you can just insert this animation into a PowerPoint presentation, and then you can use this to show your recordings in a very convenient and visually attractive format. So basically, these are all the features and most analysis settings that I described in this little tutorial video. And I would like to encourage potential users to contact me if they have some questions or issues with the program. And as I told, this is just the first part of this tutorial series, because there is the mini analysis and the spike train analysis that will be also included. Many people are interested in the analysis of synaptic currents, and I will talk about that in a separate video. Of course, there are a few other options and menus that you can use, and you will probably explore the program in detail. You can use the right mouse button to bring up the pop-up menus, and I strongly encourage potential users to explore the program using these right mouse button clicks. So thank you for the attention, and again, if you have questions regarding the program, please contact me.